Good afternoon, everybody, and welcome to the first of our new technology session. We have two sessions, one before coffee, one after coffee, in which we're going to explore some of the new technologies that are rapidly emerging and with the potential to change our lives. And so today we'll be looking primarily at the technologies themselves, and then tomorrow in the policy discussions we'll be looking at some of the implications of those technologies. And our first speaker is Professor Matthew Chang, and in the interests of professionalism I've invited Professor Sir John Scale to do the introduction. Thank you.
Which is my great pleasure. Matthew Chang is Associate Professor in Biochemistry in the Yong Lu Lin School of Medicine at the National University of Singapore and Director of the NUS Synthetic Biology for Clinical and Technological Innovation. He also heads the Singapore Consortium for Synthetic Biology, which is Synergy, and has kindly come here from the Parallel Conference, at which 900 of the synthetic biologists are attending, which is running in parallel here. His research interests lie in the development of biological systems that perform programmable functions. And so the title of his talk is Reprogramming Cells Through Synthetic Biology. Matthew.
To be here today. So what I'm going to talk to you for next half an hour is, of course, synthetic biology. But I was extremely ambitious when I was preparing slides for my talk today. So I will probably be talking to you about many things, a bit about synthetic biology and how synthetic biology has been progressing in Singapore, and also how exciting development actually have been made on this island for past a few years. So synthetic biology, there are many, many definitions because it's a relatively young field. It emerged probably about 10, 15 years ago. So in fact, there are many, many definitions because we still have a lot of work to do to really define the field. But in a nutshell, synthetic biology probably can be defined as any research activity that aims to make engineering of biology cheaper, faster, and more systematic. We're envisioning biological systems that show improved functionalities or even new functionalities that is very, very useful for many downstream applications. Shortly speaking, synthetic biology can be defined as a research practice that aims to make engineering of biology cheaper, faster, and more systematic. But what makes synthetic biology the synthetic biology that synthetic biologists have been dreaming of for past 10, 15 years is shown on the following slide. We hope to implement a more systematic way of studying and engineering biology. One example is shown on this slide: design, build, test, and learn cycle. If you look around this room, everything that you see and everything you touch actually has been manufactured through design, build, test, learn cycle. Biology actually has been making great progress, but at the same time, we see that sometimes studying biology and engineering biology can be time-consuming and also extremely not cost-very effective. So we hope to make engineering biology and entire process more systematic and cheaper by implementing design, build, test, and learn cycle. In the end, we hope to make engineering biology cheaper, faster, and more systematic. Because of this potential, and also because of the nature of interdisciplinary science that really marries chemistry, engineering, biology, and also even computer science and social science. Many developed countries have identified the synthetic biology one of the technologies that can drive their respective bioeconomy in the next century. So UK and US and many other countries, they have been growing synthetic biology community for past 10 years. And we really hope that on a global stage, this international community can come together to address many global challenges that face the world through synthetic biology. So, along this line, National University of Singapore established a research center on synthetic biology about 3 years ago. Our mission was to achieve research excellence in synthetic biology, but through this research excellence, we hope to translate our science into something very, very exciting that can be extremely beneficial in our society. And we named our center NUS Synthetic Biology for Clinical and Technological Innovation, shortly SynCity. We have garnered a lot of interest and attention from industry and also academic partners. So we now work with quite a few key industry players in synthetic biology and biotechnology. At the same time, we've been collaborating with many academic institutions and also labs to accelerate a translation of synthetic biology. Very recently, in fact, about 6 months ago, National Research Foundation has established a nationwide platform for synthetic biology. We named it Singapore Consortium for Synthetic Biology, shortly Synergy. We hope to consolidate nationwide research capabilities that exist in synthetic biology and biological engineering and biotechnology. And we have been working with quite a few industry members. So over 15 industry members from all over the world actually have come to join our consortium. And in this manner, we hope to bridge the gap between academic research and also industry research to accelerate a commercialization, industrialization of biology. So those are the Synergy members. So we have all the tertiary education institutes on board, and also we have over 15 different countries, including multinational companies and startups and so on in our consortium. And then very recently, we actually have been organized a few meetings and workshops, and we are actually coming to know each other very well. And then we have identified a few critical issues that exist in the industry. And we hope to solve and resolve those issues using synthetic biology and biological engineering. At the same time, our center and also Synergy Consortium have been making a lot of efforts in education outreach and also the outreach activities to grow the synthetic biology community of Singapore. At the same time, we hope to nurture our next generation biological engineers and also synthetic biologists. So we have started a few undergraduate modules on synthetic biology at the National University of Singapore and Nanyang Technological University. At the same time, we've been training a large number of students from our junior colleges and also polytechnics through internships and attachments. And we hope to reach out to younger generations. So we actually been working with a local comic artist to make a comic book on synthetic biology targeting our primary students. I'm very happy to bring a few copies here. So if you're interested, you can talk to me after my talk. And then right now this is a great week for science in Singapore. Commonwealth Science Conference is happening. At the same time, the largest event in the space of synthetic biology is also taking place just across the street on NUS campus. About 900 people from all over the world have gathered from over 40 different countries and from 300 different organizations from all corners of the world. So we are addressing many aspects of synthetic biology. We are talking about science and also education, policy, biosecurity, and also innovation science and also investment. So we hope to really bring synthetic biology in Singapore to another level through this international meeting. And as I told you before, synthetic biology has been around for over 10 years. And we have seen quite a few exciting applications coming out of synthetic biology research from all over the world. I mean, bioproduction has been the major research pillar of synthetic biology. For example, many things that you see in this room have been produced from petroleum feedstock. But at the same time, we believe that in the near future, we can probably produce the same materials or even better materials through biology in a more environmentally sustainable manner. Bioremediation actually has been really empowered by synthetic biology. We've been trying to make new types of drugs and also cell therapeutic regimens through synthetic biology. We also try to balance the microbiome homeostasis through synthetic biology as well. So this slide actually sums up some of the applications that are actually happening right now through synthetic biology. But stay tuned, more exciting development are on the way, and that can really solve many societal challenges that actually face this world. So as I told you before, NUS established a research center in synthetic biology named SynCity. And we have 6 research thrusts ranging from bioproduction and therapeutic cells development and waste recovery to artificial genome building. Bioproduction, as I told you before, we can produce a range of chemicals and materials using biology. What we can do is we can modify some of the metabolic pathways or enzymatic pathway that naturally exist in the nature. And by doing so, we can reprogram biological systems, including cells, proteins, and enzymes to produce our target chemicals. Those chemicals could include industry chemicals, specialty chemicals, even drugs, medicine, or even nutraceuticals. At the same time, we can build artificial genomes. Now, DNA synthesis price has been dropping rapidly for past 20 years. In fact, for past 15 years, the DNA synthesis costs have been reduced 200-fold. Now, many labs around the globe can afford to synthesize DNAs from the designing stage. We are running a few research projects in collaboration with other universities around the globe. We are now trying to better understand how genomes work by building artificial genomes. At the same time, we have one project in our SynCity that has been conducted in collaboration with our local waste management company. We are trying to come up with a biological process where electronic waste could be effectively converted into precious metals. This extraction, metal extraction, of course, has been around for many years, but this technology has been relying on the use of toxic chemicals. But we hope to do this conversion using biology and also enzymes. And we are also making a range of genetic biosensors. We can use these biosensors to detect disease biomarkers and also environmental toxins. We can embed these genetic materials onto a paper-based biosensor or even chip-based biosensor, which can be produced at very, very low cost. At the same time, we have a lot of cells in our body, and we can reprogram some of our microbes that naturally exist with us so that they can detect disease symptoms and also counteract, which is the main focus of my talk, my research for the rest of my talk today. As I told you before, we have a large number of microbes in our body. And one research question that my lab has been trying to address is whether we can reprogram our own microbes or probiotic microbes that actually show some health beneficial effects to the body to sense, detect, and even cure and treat, even prevent human diseases. Disease conditions could include infections, inflammation, cancers, even metabolic disorders. And then we can reprogram our own microbes to be able to perceive these conditions and then show some therapeutic functionalities. Those functionalities could include anti-infection activity or even anti-cancer activities. If you zoom in this research thrust, of course, there are many, many molecules that can serve as input signals. Disease markers, even abiotic signals, including pH, even nutrient gradient and growth phases, can be perceived by microbes through reprogramming. And then at the right time, at the right location, we can reprogram microbes to produce therapeutic molecules or even show some customized cellular behaviors. Of course, this research strategy may sound very straightforward because input and output and perceiving these input signals. But at the same time, of course, our body in in vitro setting has an extremely dynamic environment. If you look at our gastrointestinal tract, which is probably 7 to 9 meters long, it's very dynamic. Something keeps moving. pH fluctuates along the gastrointestinal tract, and also bile source concentration changes, and also even carbon source and its concentration vary as well. Even target sites, small intestine and large intestine, and also even some small villi. I mean, from our experiment, we have found that some microbes, they actually tend to colonize at the top of the villi. Villi is almost 1 millimeter long. And some other microbes actually tend to colonize at the bottom of the villi. Can we control this environment? Engineering aspects. Of course, for bioproduction application, higher concentration, higher production level are always good and better. But at the same time, in an in vivo setting, the higher sometimes indicates detrimental. So we actually have to make sure that our microbes and reprogrammed microbes behave within an optimal range. At the same time, once they go into the in vivo setting, there's no way to control the behavior of those microbes. Therefore, they have to act in an autonomous manner. And how about the interactions between the reprogrammed microbes and the rest of the microbial population in our body? And how about interactions between reprogrammed microbes and the host cells? So there are so many technical challenges and technical questions that we have to ask and we have to address to really realize the potential of microbiome engineering. So some years ago, probably 7 or 8 years ago, we actually embarked on a few interesting projects along this research direction. So our first target was a human pathogen, and we reprogrammed probiotic microbes to be able to sense signaling molecules naturally coming from our target pathogen, and then eventually, upon the detection, and then producing antimicrobial molecules that are very, very specific against the target pathogen. To help you understand how our system works, I brought you a very, very short animation clip. This is our target pathogen producing some signaling peptides and molecules. And we reprogram probiotic microbes so that they can intercept signaling molecules coming from the pathogen. And this interception activates the production of the killing molecules. And at the right time, at the right location, these proteins start making some holes to the membrane. Yes, this is the right timing. And then these killing molecules get into the environment and eventually reaching the target pathogen and killing them. So to make a long story short, using this model, actually we confirmed the very high killing efficacy against the target pathogen. Our killing molecules were optimized and customized and engineered to be very, very specific against the target pathogen. And also because of this sensing mechanism, all this system gets activated only upon the detection of the target pathogen. And then afterwards, we wanted to add more therapeutic functionalities to our microbes. We wanted to reprogram how cells move and how cells swim. in this particular project, we actually reprogrammed the motility, the mechanism of the probiotic microbes, so that when microbes detect signaling molecules coming from target pathogen, they start swimming towards the target pathogen. And in this manner, we actually confirm that our cells can show the directed movement towards a target location and pathogen. And in this manner, we confirm the increased killing efficacy. So we are now testing whether this system can be translated into in vivo model. So actually, we've been working with a few collaborators from the States. We are now testing whether this directed motility can really increase the killing efficacy in our in vivo model. So we wanted to test whether this system can be also, the activity can be sustained in eukaryotic infection models. So we infected worms here, and then we introduced our reprogrammed microbes. And then as you can see here, survival rate was increased from 15 to about 75% in the presence of our reprogrammed microbes. And then we actually wanted to test in animal models. We infected the mice. So at the same time, we wanted to see whether our system can lead to preventive and treatment effects. Therefore, in the first model, we infected mice with the target pathogen, and we already introduced our microbes. And then we actually confirmed about an 80% clearance of the target pathogen in mice models, and also about 98% preventive effect in the presence of our reprogrammed microbes. So far, what I have shown you is a lot of possibilities. We can reprogram probiotic microbes or even commensal microbes so that they can detect and respond to disease conditions and symptoms, and then they can counteract the target diseases. So far, the focus of my talk has been on infection issues, but we actually have some other types of microbes that show anti-cancer effects and also anti-metabolic disorder effects as well. One of the ideas that we had was maybe we can make use of some of the metabolites or chemicals naturally existing in our body. If you look at our gut, it actually contains a lot of interesting metabolites. And then through the literature, actually we confirmed that some of the metabolites that naturally exist in our body actually can serve as precursors to active drugs. In this particular project, we actually focused on a chemical, the particular type of chemical called glucosinolates. And glucosinolates actually can serve as precursors to sulforaphane, which actually shows anti-cancer effects. But our human body actually doesn't contain an enzyme that actually converts this precursor into the final active drug. So what we did in this particular project, we reprogrammed commensal microbes and probiotic microbes so that they can dock onto the colorectal cancer in the gut. And then, in a very localized manner, they actually start converting the metabolites into anti-cancer compound. To test where our hypothesis will really work, actually we conducted a series of experiments. In this experiment, we actually confirmed the direct binding between reprogrammed microbes and cancer cells. At the same time, in the presence of broccoli, so that glucosinolate chemical class actually comes from broccoli and other types of cruciferous vegetables. So we fed mice with broccoli extract and water containing our microbes. And then in vitro setting, we actually confirmed the killing of the cancer cells in the presence of broccoli extracts and our reprogrammed microbes. So we induced a cancer in mice, and then we fed the mice that actually contain a colorectal cancer with reprogrammed microbes and broccoli extracts. And then after 16 weeks, as you can see here, our enzymatic conversion took place in vivo setting in mice. Therefore, we confirmed about 80% clearance of the colorectal cancer in the presence of reprogrammed microbes and broccoli extract. So this is another possibility, because our human body gut, skin, even upper respiratory tract contained a large number and classes of chemicals and metabolites. Reprogrammed microbes can sense, and they can go, and then they can treat target diseases in vivo setting. But as I told you before, so I've told to you about a few types of commensal microbes that have been reprogrammed to detect and treat some target diseases. But at the same time, as I told you before, we also have to implement some control elements in our microbes so that they can detect and they can counteract and prevent and treat the target diseases in an autonomous manner. So what we did in this particular study, we actually made a switch, genetic switch, that can be activated by a specific target chemical and also by the availability of carbon sources. Our large intestine doesn't have much glucose. Therefore, we thought that this actually can serve as one of the input signals that can decide the corresponding action of the reprogrammed microbes. So we actually made a genetic controller that comprises a switch. So only in the presence of both target signals, this genetic switch gets turned on, therefore downstream processes and functionalities can be activated. And in this manner, we confirm that cells become happier, our microbes become happier because they don't actually have to produce enzymes and the implemented genetic controller all the time. Once they reach the target location, and also once they reach the target the site, and also once they meet the target pathogens or symptoms, entire genetic circuit can get activated. Therefore, they can preserve a lot of metabolic resources. Therefore, cells have become happier. So they actually undergo much less metabolic stress, and then they can really pinpoint the location and timing of the activation of the enzymatic conversion. Another example is a pH. Our pH in the intestinal tract actually fluctuates. So pH actually can serve as an input signal that can decide the activation of the implemented genetic circuits. So in this particular project, what we did was we actually made a genetic pH sensor. To make a long story short, what we did was we actually took a natural pH sensor and then we rationally redesigned and then we optimized the response range. Therefore, we actually reprogrammed the pH sensor to turn on and off at our target pH ranges. And we also optimized RNA polymerase because the efficiency was too high, therefore the separation was not very optimal. Therefore, we actually re-engineered RNA polymerase to optimize our transcription processes. Therefore, in the end, actually, we confirmed the large separation by the target pHs of our interest. And then what we did was we actually used synthetic biology biofoundry. Synthetic biology biofoundry is a small biological engineering factory that can be empowered by a series of automation and high-throughput equipment. We can do screening, we can do assembly, we can do synthesis, and also we can do detection using biofoundry. So we actually made a pH genetic sensor, and that responds to our target pH changes. And then another project actually coming out of this big space is actually improving the pH tolerance of our microbes. So when we eat, drink probiotic drinks, as you know, about 99% of the cells eventually die at low pH. If we can reprogram probiotic microbes or even commensal microbes to become more tolerant against low pH, they can survive, more cells can survive this low intestinal tract environment, low pH intestinal tract environment. So we actually made a pH genetic sensor. And what we did was actually we used the mutation mechanism. So our hypothesis is as follows. If the cells are exposed to low pH, if the cells don't know yet how to respond and how to survive at low pH, intracellular pH follows extracellular pH, which is low. But somehow through mutations or through evolution, if they have come up with a mechanism intracellularly, they can make cells withstand the low pH. We hypothesize that intracellular pH still remains neutral when cells are exposed to low pH. But this is a long process, which usually takes a lot of time, but we actually wanted to automate this adaptive evolution process. Using this pH that I briefly described before, we actually put a few genetic circuits. If the intracellular pH stays low, which follows extracellular pH, then what happens, mutation just keep occurring to the genome. Therefore, we can accelerate this genome shuffling and mutation process. But when the cells are exposed to low pH, if the intracellular pH stays neutral, then what happens is this transcription direction flips, then red proteins start being produced. Therefore, cells actually turn red. We actually automate this process. So to make a long story short, within a very, very short time frame, which was only 36 hours, we managed to evolve the strains to become more tolerant against low pH. And then we actually tested our microbes against some organic acid that cause low pH. And then using our evolved strains can actually survive and grow much, much better at low pH. So this is actually a glimpse of what synthetic biology-driven adaptive evolution can really achieve. We really shortened the time required for adaptive evolution from several months to only 36 hours using this automation process. So today, actually, I brought you 4 distinctive examples of what synthetic biology can offer. Microbiome engineering, at the same time, we can implement some control elements in microbes so they can behave, they can sense, perceive external conditions, and they can act in an autonomous manner. So we are now trying to push the boundary of synthetic biology. And as I told you before, you will see more exciting developments coming from synthetic biology in the near future. I want to end my talk to just reiterating how important all the collaborations between Singapore and other members of Commonwealth. I mean, of course, UK has been the big part. I often say that without UK support, there would be no synthetic biology in Singapore. Paul Freeman and Richard Kidney and many others, who are actually here on NUS campus right now, they really helped us map out our plan for synthetic biology. Also reaching out to many other countries in Commonwealth. We have exciting collaborations with Australia. And also we are also trying to forge some collaborations with India and also other members of Commonwealth. So this is extremely exciting in the community, and which has an extremely enormous future and potential where synthetic biology can be seamlessly translated into something very exciting that can really change how we live and make an impact into our daily lives. With that, I'd like to thank you for your attention, and I really believe that if we come together, we can really build a better world together. And thank you for your attention.
Other questions for Dr. Chang? not difficult. It's difficult to see from here, so you should stand up if you have a question, I say.
And also, if you need any the comic book, just talk to me after.
In your probiotics against the cancer, I didn't quite get it. Is it necessary for the probiotic bacteria to associate, to interact with the cancer cell?
Oh, no, actually, those are new functionalities that we designed and we built in probiotic microbes. I mean, probiotic microbes don't naturally just bind to colorectal cancer.
No.
But we actually engineered the membrane structure of the probiotic microbes so that they can actually target certain receptors that exist on the colorectal cancer so that binding can take place.
So those are specific receptors on the cancer
specific
cells.
receptors. We actually tested for other types of cancer, but very, very specific towards colorectal cancer.
Right. Okay. Interesting. Are there other questions for Dr. Yes. Do you see that?
I have a question about your pathogen killing experiment.
Where are you?
Right here?
Yeah.
Hello.
Right at the back.
Are you worried that, so this was, you used E. coli to produce a compound like a pyocyanin of some sort
Yes.
against Pseudomonas. Are
Yes.
you worried that Pseudomonas would become resistant to that?
Eventually, that's a possibility. But at the same time, antibiotic resistant issues is really big. At the same time, it might be actually, we really have to diversify our antimicrobial strategies. So far, we have relied on the chemical antibiotics to treat infections, but at the same time, these kind of mechanisms or some other mechanisms that we can come up with, we can actually implement all this together. We can probably come up with a solution for the emerging antibiotic resistant issue. Yes, going back to your question, yes, that can happen. But at the same time, so far, we haven't used pyocin a lot for anti-infection application. So we haven't seen any emergence of the cellular mechanisms against pyocin yet. But of course, we cannot just rule out the possibility of seeing resistance against pyocin in the future.
Okay, Anna Kranvis from South Africa. What are the ethics of synthesizing new genomes?
Right, yes. That's a very, very good question. So right now, we are just trying to understand how genomes work by building. So a potential application, because we're actually using this system for Saccharomyces cerevisiae. Saccharomyces cerevisiae actually has been used for thousands of years for a range of biotechnological applications, wine, beer, bread, and so on, even drugs. So we are trying to better understand how genome that comprises 16 chromosomes works in Saccharomyces cerevisiae using artificial genomes. But at the same time, yes, if we actually want to really bring this to the application stage, we actually have to really engage public. We actually have been talking to the public. So we actually have organized many, many workshops that try to address these ethical issues. But we actually haven't come up with any applications yet from that particular project. But in the future, if we really take this to the commercialization or industrialization, translation stage, yes, we really have to address potential ethical issues.
Any more questions for Dr. Chang? Yes, there's one there.
Hi, I'm Carl from New Zealand. My question is, rather than antibiotics, have you considered getting the bacteria to produce bacteriophages?
Yes, yes, it is one of the ongoing projects in the lab right now. So we are trying to make use of some other cellular mechanisms, including phage infection and even conjugation, and also even CRISPR. We are trying to embed all these mechanisms to identify the most optimal antimicrobial strategies based on the system that I described today. Yes, we have actually a few ongoing projects based on phages.
Okay, at that point, thank you again.
Thank you.
Thank you very much for coming.
Good afternoon. My name is Bill Wakeham from the Royal Academy of Engineering in the UK, and it's a pleasure for me to introduce a fellow engineer to you this afternoon, and that is Professor Peter Corke. One of the technologies we felt would have a profound effect upon humanity in the future would be robotics. And Peter is Professor of Robotic Vision at Queensland University of Technology in Australia. He obtained his first degree in electrical engineering from Melbourne, and since then his work on robotic vision has earned him an international reputation. He is the director of the Australian Centre for Robotic Vision, and not only has he built a sound reputation for his research, but actually he's been internationally recognised for his contributions to teaching in that area through efforts with MATLAB and the robotics toolbox therein, and also for a very well-respected textbook. So for a very great pleasure for me to ask Peter to come and talk to you about his interest in robotics as applied to things outside in the field.
Thanks very much, Bill. It's my pleasure to be here today. And what I want to do is talk about robotics in agriculture. We're talking about an emerging technology being combined with a very important and very ancient human activity, agriculture. Another way we might think of this is about artificial intelligence and food production. And I want to start by trying to unpack artificial intelligence. Many of you in the room have probably got an idea about what artificial intelligence is. There's lots of scary headlines at the moment. Stephen Hawking and Elon Musk banging on about the dangers of artificial intelligence. I think there are very real dangers down the track with artificial intelligence, but I think it's beyond my lifetime. We've got a long, long way to go. So I'm going to start, as I said, with artificial intelligence. Artificial intelligence is a very, very old research activity. It can be traced back to his seminal event, the Summer Research Project in 1955, with some very, very bright people. So we got McCarthy, Marvin Minsky, Newell, Simon, and others like that. In 1955, they got together for a summer at Dartmouth College and put together an agenda that really shaped artificial intelligence. And their focus at that time was computers were just starting to become available. People were excited about this. And they were thinking about neural networks. Even back then, they were thinking about neural networks. Natural language processing, theory of computation, games was a really big early driver of artificial intelligence. Interesting, robotics is not here. The first robots didn't really start to appear in factories and research labs until the 1960s. So the AI agenda predated robotics. And there's an important difference between AI and robotics. And the way I think about it is AI is about reasoning, it's about moving information around inside a computer system or a computer network. A robot is an AI that does physical work. It's an instantiation of AI in the world that we live in. It can move around, it can pick things up, it can put them down. That, to me, is the key difference between AI and robotics. And so AI had a number of goals, and chess was a very, very early goal for the AI community. And we knocked off the best human on the planet in 1997. You know, it's more than 20 years ago now. We beat grandmasters. And just last year, very notably, we beat the world master Lisa Doll at the game of Go. So things that have been considered by humanity as incredibly difficult, you know, the sort of pinnacle of human intellectual endeavour encapsulated in these games, computers can do that now. More recently, we have very low-end products, Amazon Alexa, Google at Home, whatever, little microphone thing that sits in your house and picks up what you're talking about, and if it thinks you want to buy something, it'll hit up your credit card and buy it and have it delivered to your house. This is an outstanding achievement in sort of commoditizing speech understanding and natural language understanding. Very, very early goals of the AI movement. It's now a product, you can buy it for $100 or something like that. So we've come a very, very long way in the AI in the AI movement. But here's the thing. Computers can beat chess grandmasters and grandmasters at the game of Go. The kid on the right-hand side here who's picking up a chess piece is beyond our current ability. And that might sound odd to you. Robots don't have the ability to be able to look at shapes in a very robust and reliable way and say that's a pawn, and nor do they have good hand-eye coordination, nor very good manual dexterity. So without using almost no conscious effort, I can reach over and pick up a water bottle just like that, quickly, reliably, every time I'll pick up the water bottle. Robots still can't do that. It sounds stupid, but an 18-month-old child has got better hand-eye and dexterity capabilities than robots do today. Lots of people are working on it. It's an important problem in robotics, but we've got a way to go. So this is encapsulated in a paradox that's attributed to Hans Moravec. Things that humans find hard, computers find easy. Things that humans find easy, computers might find hard. So here are the examples of state-of-the-art robotics. On the left-hand side, we have robots building Tesla motor cars. These robots are very fast, very strong, very reliable, work 24/7 and don't complain. They can pick up bits of cars and put them and connect them to other bits of cars. They really don't know what's going on. They can only pick up the things that they're designed to pick up. We custom design their hand to do the job that they need to do in the factory. They're not general purpose. Not like this is general purpose. We custom engineer grippers for robots. And they can't see. They really don't know what's going on. And the ones on the right-hand side, these are Amazon dispatching robots. So these carry shelves of product around Amazon fulfillment centers. Amazon own a fleet of 30,000 robots. It's what enables them to be the fast, efficient, cheap shipper of merchandise that they are. They are a robot-enabled business, perhaps the first one on the planet. But all of those robots that I just showed you, they can't see. They can't see like we see. So here are some examples of people doing seeing. So the girl's looking at the Rubik's Cube. She's building an internal mental model. She can reason about it. She can adjust the cube to solve it. The guy in the middle's juggling. He's using his eyes. He's got an internal dynamic model of how things move in a gravity field. And he can predict where they'll be in the future. And arguably, that's the great advantage of vision, is it allows us to predict what will happen in the future. Because I can see things that are a long way away before they're going to hurt me. I can predict what they're going to do in the future, and I can react. That's got an enormous survival advantage. Vision is a very, very powerful, important sense of humans that most of us take utterly for granted. And we can use it for things like driving, the picture there on the right-hand side. Vision's such a good thing that evolution invented it not once, but 10 times, right? It's been on the planet for about 700 million years. It appeared at the beginning of the Cambrian explosion, when life got interesting on planet Earth. Before that, life was really boring on planet Earth. It became really interesting. There is an argument that it was the advent of the sense of vision that caused the Cambrian explosion. And there's some interesting papers on that particular topic. So there's 10 different eye designs on the planet. 96% of species have eyes. It is the best way we know of, evolution knows of, to understand what's going on around you. Absolutely invaluable. But it's wickedly hard for robots. And I'm going to try and just give you a few examples to hammer home why vision is such a hard thing. Okay, you look at that. And he's not a little guy on my finger. It's clearly a guy who's taller than me, but he's further away. And when we look at things, there is this coupling between distance and size. And that's a function of the way that images are projected onto the retina of our eyes. We take a three-dimensional world and we crunch it down to a two-dimensional image. There's collateral damage. You can't just throw away dimensions. And that what happens is that we conflate scale with distance, right? And this is a really big problem for a robot. Is that a little guy or is it a big guy in the distance? We cannot tell. There's not enough information. The problem is ill-posed. Here's another one. Left and right columns are the same place. And we can see in the top row, well, it looks like the same place to us. It looks like the same bridge. We can see the same trees in the background. But one's blanketed in snow and one's not. So they look, the appearance is radically different, yet it's the same place. Try and write a computer algorithm that will tell you that's the same place when they look so different. The bottom example, It's the same place, but one's covered in shadows. Shadows are a natural part of the world. Our brain just filters them out. Write an algorithm that can detect that that's the same place despite the big change in illumination pattern. It's very, very hard. This is a cup. You all know that that's a cup. But each of these cups looks different. They're different shaped cups. They're cups taken from different viewpoints, cups that are empty, half full, full. Try and write an algorithm that can tell you that that's a cup reliably under all different lighting conditions, viewing angles, and whatever. We're making progress on all of these problems, but we're far from having robust, fast solutions to all of these. So, just trying to paint a bit of a picture that robots, in the sense of vision, it's not what you read in the newspaper necessarily, right? There are still really hard problems. We've got a long way to go. So we're going to talk about food production. And I'm not going to lecture people in this room, but the root cause of problems with food production comes down to this graph. Too many people on the planet, now and into the future. And the consequences of this is we're going to need more resources, we're going to need more transportation to move all those people from A to B, and we're going to need more food. Robotics has got a role to play in all of these. There's a lot of interest, a lot of work happening in robotics for resource extraction, for mining. Mining involves putting human beings in very dangerous and unnatural places, deep underground. Humans shouldn't be there. Robots can do that work. Transportation, you've probably all read about Google cars, autonomous driving projects, Nutonomy here in Singapore, Oxbotica in the UK. Lots of interest all around the world in self-driving. I'm going to just talk a little bit about food. All right. We produce, at the moment, enough food to feed most of the people on the planet, and that's been underpinned by a number of revolutions. Genetic revolution, mechanization, and herbicides and pesticides. It's allowed us to feed all the people on the planet. And this graph from the United States shows that we can produce the food we need with a tiny fraction of the workforce we used to need. So over 200 years, the number of people we need to produce the food to feed us, or probably overfeed us, has been dramatically reduced. And so here's some examples of mechanization. We've moved from using human labor to animal labor to steam power to diesel power. And the machines have gotten bigger as we've gone along. A consequence of making the machines bigger is that the pressure these machines exert on the ground has gone up. Ground pressure goes up. And what that means is we squash the ground upon which we drive. And that damages the root systems of the plants that grow underneath where we drive. Big problem today as machines get bigger and bigger. And the solution to it is to use GPS-guided vehicles. So they go up and down the field and crush the same piece of ground over time. So they don't crush the whole field, they just crush particular bits of the field to an extraordinary degree. And we tolerate that. But it might take 1 to 1.5% of our land out of production because we've crushed it under the wheels of great big machines. As ground pressure goes up, the number of days in the year in which you can drive a big machine on the field goes down. If the ground is soft, you can't put a big, heavy machine on the ground. It will sink. Weeds are a really big problem in agriculture, and my talk is mostly around robotic weed control. In my country, we apparently spend $1.5 billion a year in weed control, and the industry estimates that it's costing it nearly $3 billion a year in loss. We don't like weeds because they take moisture and they take nutrients that would otherwise go into our crop. The weeds compete with crops, therefore we don't like them, therefore we want to eliminate them. And one of the best ways to eliminate them that we've come up with is to spray them with herbicide. Lots and lots of herbicide. And we've been doing this for decades. We build bigger and bigger machines to spray huge amounts of herbicide onto their crops. It's the agricultural system that we've built. It's industrial agriculture. It works, but it's certainly not optimal. And you might ask the question, why are we building tractors that are bigger and bigger and that go faster and faster? Why do we do that? And the reason we do that is because of this thing. Come on. The driver, right? We only need to have a really big machine so that we can make the driver as effective as possible. He can get the most work done if he's in a machine that covers a lot of ground very, very quickly. And as a roboticist, I say, what's a driver? Why do we need a driver in the 21st century? There's a lot of automation starting to happen in in agriculture now. So this is a kind of fairly ordinary tractor cab now, and it's bristling with equipment, GPS guidance. There's generally still a guy sitting there kind of surveying and just making sure that it's all working nicely, but the actual driving is now ceded to a machine. It's very expensive, this. And we're starting to get farms now that are quite complicated. They depend on satellite navigation systems, base stations, on-farm transmitter systems. We've got building really complex technological systems on our farms in order to navigate these machines across the ground. Maybe there's a better way. And so what we're interested in our project is saying, if we don't need a driver, then the machine could be smaller. And if we could get the same amount of work done by having lots of machines working together in parallel. Instead of one big machine, you have lots of little machines. The machines don't crush the ground, they don't need drivers. There's a lot of advantages in going this particular way, and the technology should allow it to happen. So this is the first prototype that we built in 2014. It's speeded up by a factor of 8. You can see it running backwards and forwards there, navigating, avoiding obstacles, and doing the sorts of things that a human-controlled tractor would do. So how does this stack up? The traditional approach with a great big machine with a person in it, or a smaller machine? So here are some statistics. Conventional machines may be 30 meters wide, goes 20 to 30 kilometers an hour. It's got some disadvantages. If we built a robot, it wouldn't be as wide and it wouldn't be as fast. So all up, it's 1/24th as productive as the big machine. But if its cost was 1/24th of the big machine, and I bought 24 of them, then I'm where I was, right? I've now got a lot of modular robots instead of one big robot. And if one of the robots breaks, I'm fine, I've still got 23 left. If my big robot breaks, I'm in very big trouble. So there's a robustness issue that comes with going modular. So we've got to get the cost down. We've got to get the cost of the robot down, and we've got to get rid of all this kit. This is tens of thousands of dollars worth of stuff. We've got to throw it out. Can't afford it. What are we going to do? The human being who drives the tractor looks out the window, and he sees this. He drives the tractor. So can we do that? So this is the challenge behind this particular research project that I'm sharing with you. And the motivation here is, can we just do this with a camera? We know a human being can do it just with a camera, well, 2 cameras in the case of humans. We know the cameras are really cheap. We've all got lots of cameras. We've got cameras in our pockets. Apple probably buys them for $1 each or less. Cameras are cheap. Computers are cheap. What can we do? So this is the prototype I showed you before, and just sort of as a sense of scale, the little green machine is the one we built. The big red one in the background is a fairly traditional weed spraying rig. So there are 2 key robotic vision challenges that we needed to overcome. One was the ability to follow crop rows, and the other was to avoid obstacles. This is what the human being does. So I won't delve into this too much, but we created an image processing pipeline that takes images, does various things to them, and generates the steering commands to the vehicle. Keeps it parallel to the rows and centered on the rows. And that can be done with fairly modest amount of computing from just a single camera. And there it is. There's overlaid the tractor path on top of Google image of the field. It does a very good job of row following. And the other capability it needed to have was obstacle detection. So anything that's in a red box here is something we've detected as being an obstacle, something we should try and avoid driving into. And that worked quite nicely. And in here we see Dave Ball, the engineer who led this project, very confidently standing in front of the robot that he built, and it avoids him very nicely, moves around, and then it's trying to get itself back onto the original crop rows, get back driving along the line that it was originally following. So it's an example of a machine that can see. Not nearly as well as we can do. All this can do is follow crop rows. Human beings, amazing general purpose vision systems. This is a very specific vision system, but it's starting to push the edge of the envelope on what robots can do. We got some more funding. We built a much nicer robot, which you can see here, a very beautiful robot, and you'll see more pictures of this robot doing interesting things in a moment. So now we've got a robot that can drive along a crop row. That's great. It's not going to drive over the crops. It's going to drive parallel to the crops and keeps its wheels in the groove. Now we want to control weeds. So can we detect a weed? Can we look at a plant and say whether it's a good plant or a bad plant? And with developments in deep deep neural networks, convolutional neural networks, we can train a network that can recognize a good plant from a bad plant. So with down-looking cameras, the robot's driving along, it's looking down, looks at each individual plant and says, good plant, bad plant. This is quite easy to do. So now the robot's got a high-level cognitive function here. It's doing what a human would do if the human was walking along the crop row. And once we've done that, then we can start to do very, very targeted spraying. Not just spraying willy-nilly, where a large percentage of the spray goes into the air, into the environment, just on the plants. We can reduce the amount of herbicide that's required by an enormous amount. And this is a little bit busy, but we think that a fleet of multiple modular robots doing targeted spraying could reduce the cost of weed control, chemical weed control, by 90%. The numbers are astounding. The technology is doable. We can do this today. But the weeds strike back. We've been pouring chemicals on weeds for 5 decades. And just like bacteria who learn to become resistant to antibiotics, the weeds have become resistant to the herbicides of choice, glyphosates. And around the world, people are becoming very alarmed at the rise of herbicide-resistant weed species. The problem's very big in the US, where they've been doing it the longest, big in Canada, big in Australia, growing in China. There are very alarming infographics here that I found about some amazing weeds that grow very quickly and produce millions of seeds. This is a worldwide problem, and people are really worried about it. So if you can't poison the weed, what can you do? Well, you can get more exotic poisons that are more expensive and more noxious that you don't actually want to have in the environment, and that's a way people really don't want to go. We can then start to do mechanical cultivation. We can have arrays of flamethrowers. We can do microwaves. This one in the bottom right corner is an array of microwave antennas. As you go along and you just nuke the weeds, and they wilt. Like you wilt spinach in a microwave, you can wilt a weed in the field. So lots of ingenious people thinking about different ways of controlling weeds in ways that we hope they won't develop resistance to. If they develop resistance to this, we're in deep trouble. So this is the next addition to our weeding robot. And here what we're doing, instead of before we saw a weed and we would spray it, now what we do when we see a weed is we dig it out, chip it out of the ground. And that's very nice. No chemicals involved now. The machine is entirely electric. It's using electricity for propulsion. It's using electricity for doing sensing and analyzing the scene. It's using electricity to eliminate the weed. So this is what our ancestors have done. We're doing it now. It's the same approach. It's individualized plant treatment, but now it's being done by an IT system, an AI, rather than by people. I want to talk very briefly about horticulture. Horticulture is basically the production of fruit, vegetables, nuts, and flowers. That's the textbook definition of horticulture. In my country, we have a labor problem in my country. It's not that everybody's employed, it's that lots of people don't want to go and work on farms and pick fruit because it's hard work. And the coffee's bad and the internet's slow. So people don't want to go into the country and do this work. So we get European backpackers to come and pick fruit for us, and that sort of works. And we also have special visas for Pacific Island nations to come and pick our fruit because Australians don't want to do that work. So we were talking to people who produce these, and we call them capsicums, maybe you call them bell peppers, I'm going to call them capsicums. And so this is a big crop in Australia, and we've done quite a bit of work on identifying them on the vines. Some of them are quite easily seen, some of them hide behind leaves, so we're using a mixture of traditional colour imagery and also near-infrared imagery to find the fruit. And then we created a robot, the robot's called Harvey, and Harvey's job is to separate capsicums from the vine on which they're growing. So there he is. It's a wheeled base. It's got a vertical lift axis and a robot on it. On the end of the robot is a tool that can attach to a capsicum and sever it from the vine. So he's getting a bit of a look. He's moving around. He's going to build up a three-dimensional model of the fruit. And in the three-dimensional model, three-dimensional structure, color, texture, he can work out what's a capsicum and what's a leaf, figure out the best place to attach to it, moves in, attaches to it with a suction gripper, and then cuts the stalk. And I have a capsicum in my hand. At the moment, he's slower than a human being. He started off about 8 times slower. He's now up to 2 times slower than a human being. He's getting to the edge of commercial viability because this robot can work day and night. It's not going to complain. And it's doing a pretty good job of recognizing the fruit and removing them from the vine. So this is a very, very nice project. We chose the capsicum because it's a robust fruit. It can tolerate quite a bit of abuse. This would be too harsh for a tomato, and it was not going to work for fiddly little fruits like berries or something like that. There's a lot more work needs to be done. But there's people all around the world looking at robots for the different kinds of fruits and flowers and nuts. Lots of work in this field. So I'm going to finish there with a few simple take-home messages, right? Robots are real. They're not like the things you see in Hollywood movies, right? They're real, they're functional, they don't look like Hollywood robots, they're not as good as Hollywood robots, they're not as good as C-3PO or R2-D2 yet, but we'll get there. Their visual competence and their hand-eye coordination is still very poor. So we can engineer some solutions for some specific cases. We don't have a general theory of robotic vision or robotic hand-eye coordination yet. We will get there. But I think what's interesting to me is that agricultural robots offer the possibility of treating plants as individuals. We lost that when we just sprayed them en masse. We said every plant is the same. We're going to put the same dose on it. And that's absolutely not the case. A robot could go along, could look at each plant, could look at the leaves, figure out if it's being eaten by a beetle, squash the beetle, or do something else. You don't have to do the same thing to every plant. It's an intelligent system. It can take plant-specific action. And it may be one means to try and meet the food needs of a growing population. Thank you very much.
Peter, thank you very much indeed for a fascinating talk. We've got plenty of time for questions, so if there are any, please.
One up there.
Yep.
Where are our microphone runners?
Microphones? On the right, on my right side, your left side.
Anthony Dodge, University of Bristol in the UK. Just like plants evolve biochemical resistance to herbicides and other chemicals, plant morphology evolves quite quickly also. Mm-hmm. And with a robotic system that's based on visual recognition of plants, one might envisage that you would get some evolution of the shape of weeds in order to escape detection. And I'm wondering how rapid or easy it is to reprogram the visual recognition of weeds in order to accommodate those kinds of changes that might occur through evolution or because of different soil types in different parts of the country.
It's a really good question, and it hadn't occurred to me that plants may dodge the bullet by changing their appearance, but animals have done that for a long time. The tool that we bring to bear on this at the moment is a machine learning technique. So we give lots of examples of the weed, lots of examples of plants, and they're labeled data. We say this is this kind of weed, this is weed X, this is weed Y, this is a good plant, and we train the network on that. So if you can train it for one set of appearances, you can train it for a different set of appearances. It would need someone to be alert to the fact that maybe I'm going to need to retrain it someday, but we know how to do that. When we would need to do that is probably an interesting question. Thank you.
Thanks. Question over there,
okay.
please.
Hi,
and
yeah.
there's one up here as well. Yes.
I'm Sarah from the University of Washington. Thank you for your talk. I wondered what your thoughts are about the social implications of the loss of jobs with increased automation and more robots being used. So autonomous vehicles being a good example of losing truck drivers in the US, for example. What are your thoughts?
Absolutely. Job destruction is going to happen. There is a lot of very alarmist reports in the media. I guess the root cause is probably the Martin School at Oxford saying that 47% of all jobs are going to disappear in the next 10 or 20 years. Jobs will disappear. Whether new jobs come back, I don't know. I don't have the answer to that. I think what's been surprising with the job destruction is I think for a long time, people thought that if you'd been to university, you had a profession, you were immune from this. There's going to be people who did physical work who were going to be the targets of job loss. I think it's actually surprising. I think that artificial intelligence, not robots, artificial intelligence is going to cut out a huge swath of jobs, right? And that's just systems that manipulate information. So, the example that's often given is radiology. Looking at a medical image and figuring out if there's a pathology there. That's something that computers can do much, much better than humans. Grading essays at university level can be done better by an AI than can be done by humans. So a lot of white-collar jobs, professional jobs, I think are going to be destroyed before what we sometimes call blue-collar jobs. I cannot imagine a robot that could do the job of a plumber, right? What plumbers do is absolutely extraordinary in terms of the physical skills and dexterity and sensing and understanding that they need. I think plumbers are going to be the last people with jobs on the planet. And then roboticists, yeah? Roboticists and plumbers.
Right, there's a question there, and then one down here, please. Yes,
yeah, I had the similar kind of comment. Ethics part of it is one that when you use a lot in not only in agriculture but in other fields, that mechanizing the humanity to misuse of technology, maybe criminal and other activities of robotics when it evolves.
Well, there will be criminal misuse of robots. There will be cyber attacks on robots. If there are fleets of self-driving cars on the roads, people are going to find it's interesting sport to try and subvert them or do dangerous things to them. We can just build in the best protections that we can, but we know that it's pretty tough, right, to keep any kind of IT system safe, particularly if it's networked. And robots to be useful, I think, will have to be networked. It's an area that, certainly within my community, the robotics research community, security, cyber threats is not on anyone's radar. It absolutely needs to be. It needs to be baked in from the beginning. If we learned anything from computer security, it needs to be baked in from the beginning.
Thanks very much. Yes, there, please.
Michael Kelly, Cambridge. Peter, thank you very much for a wonderful talk. I was struck in particular by the selective use of the liquids to kill off the plants. Is the converse true, the selective use of water to more highly target the use in places which are otherwise water stressed? I can see it being a possibility in engineering terms, but do the numbers, economics add up?
I really don't know. It's a really good question. In a country like mine, you think that it would be, we would have thought of that before someone from the UK. Look, it's probably going to be a lot more economic than running irrigation systems. And irrigation systems, need an enormous amount of upkeep, to keep bugs out of them, to keep leaks out of them. It's a really good idea. All right, you thought of it first.
Thanks very much. Tony.
Yeah.
Yes, Peter, thank you very much indeed.
You're welcome.
What's the state of the art in applying this type of technology in mining and quarrying?
Robotic technology for mining industry has been very active for probably almost 2 decades. For the mining industry, they've got a number of challenges, important economic challenges. One is that increasingly they need to look after their workers. Once upon a time you didn't, now you do. So their workers' insurance premiums are really, really high. So they need to take people out of hazardous situations. So we can have have robots in a hazardous situation. We need to take humans out of hazardous situations and replace them with robots. So the interim step is what we call teleoperation. So that is the human sits in a control room somewhere, maybe on the surface, and radio operates the machines that they would normally sit in the cab of and drive underground. And that's an interesting first step. What happens then is, instead of having one remote driver controlling one machine, you haven't saved any labor at this point, but you've made the human safer. Now what you do is you make the machine a bit more intelligent, so it can do some of the job by itself. So it can drive for 500 meters and then turn left and then stop when it gets to the thing. So then you could have one human managing 5 machines or 10 machines, because most of the time those machines, 80% of the time, those machines are doing their own thing. They just need a bit of occasional supervision and intervention. So that improves your productivity an awful lot. So the mining companies are really interested in this. They've got huge amounts of money tied up in capital. Mining machines cost tens of millions of dollars and big open pit excavators. And if they can improve the productivity of that asset by 1%, that's a massive amount of money. The research, the technology will pay for itself. So a big interest, particularly for the people who run the mines, the people who make the equipment are kind of technological dinosaurs. They just know how to make things out of metal. They haven't figured out computers yet. And so they're going to need to evolve and lift the game to meet the needs of the miners who are really hungry for innovation.
Thank you.
Mr. Red, question over there, please.
Yeah, Rashid Smiler from the University of British Columbia in Canada. Yeah, as you were describing the increasing size of machines on land, I was thinking of the ocean, and we see a similar increase in fishing boats getting bigger and bigger. The Atlantic Dawn could just zap up all the fish that people catch in a year. And the reason you gave for this, the driver, right? Mm-hmm. I'm wondering whether that will also apply to the ocean. Economists will talk about increasing scale in general. So how does that work in here?
I think for ships, you don't have the constraint that we have on land. I mean, on land, we have the constraint of crushing the ground. For big ships, you don't have that constraint. And already these big container ships have got very, very minimal crews. And there is already talk of automated container ships that have got no crew at all. Maybe have a satellite telemetry link, they're GPS controlled, and the ship just pings you every hour and says what it's up to. It's robust to piracy. It's got a lot of advantages. This is a technology that will come. What it means for fishing in particular, I don't know. But certainly to make big boats with fewer crew is technologically possible.
Well, I think, there's one last question up here, please. Wait for the microphone, please. It's coming.
It's just there.
Okay. I'm Satinder Kaur Brar, INRS Canada. My question is there a possibility to link drones to these ground robotics so that prevention and treatment can go hand in hand and we have precision agriculture, which we're always looking for?
There's a lot of interest in this at the moment, what's called weed scouting. So you send out a drone, and the advantage of a drone is that it can cover a lot of ground very quickly. It takes images, it figures out where the weeds are, and then the slow lumbering ground machine goes out and does away with the weeds. There's also been work on using drones to apply chemicals to crops. But the disadvantage of that is that the payload capacity of drones is very, very low. So they can't carry very much herbicide or pesticide on board. So this hybrid that you alluded to, we have drones out there scouting the environment, then the robots plan what they're going to do and go in there. I think it's a very good one. It will come.
Okay, thank you.
Thank you, and thank you all very much for that. And Peter, thank you very much for your talk. I'm sure everybody will want to join with me. Thank you.
Is it a break now? Okay. Two and three.
Yes, actually, we have the coffee break now. So the coffee will be level 2, and which is the sort of lower level here, and level 2 up at the top. So I think it's back in half an hour. Leslie? Is it back in half an hour? Thank you.